Great. Well, thank you for the introduction. I am Devin Lange, a PhD student from the University of Utah and a current member of the Visualization Design Lab. Assuming things go well, that's only for 2 weeks more. And after my defense, I will be starting at the Harvard Medical School with Niels Gehlenborg. And for my first talk today, I will be discussing some work from my internship at OZ Technologies. So let's get into it. Maybe. If I can. There we go. Great. So let's say you have a population of cells and you're trying to characterize them in some way. Maybe you want to compare these with other populations of cells. One way that you can do this is to do single cell annotation. There's a broad category of techniques that involve looking at each individual cell and recording some attribute of that cell. And the visualization techniques that I'm going to be talking about today really applies broadly to this general single cell annotation. But to make it specific for this talk, we're going to use the example of looking at surface proteins. So all of our examples are going to look at cells that have their surface proteins recorded for each cell. And I'm not going to talk about how we collect this data, but some of the founders of OZ have some nice literature on some of their techniques for doing this. So you have these biological markers, these surface proteins, and can use these to define a cell type or phenotype. So the second row that I've circled here would be a cell type or phenotype where CD3 is present in the cell, CD4 is absent, it is not there, and CD8 is present. So this would be like a CD8 T cell. And if you look at your biological sample, you can count the number of cells that fit into each of these phenotypes. So here we have 3,898 cells that are these CD8 T cells. And instead of printing the numbers, I'll use colored for the rest of the talk. And of course, this is one sample as a column, but you can have multiple samples here, right? And for each sample, you can record different metadata relevant to your experiment. That could be blood type, or if it's in the arm or cohort, or really anything here. And already we've constructed sort of a nice visualization for this use case. Here, it's easy because we have only 3 phenotypes and 4 samples. So the challenge is what happens when you have many more markers that you can distinguish. And this is the challenge that comes with OZ's technology. Because they have the ability to distinguish so many markers, you end up with thousands of these phenotypes. And this is really powerful to be able to collect this information, but it's a bit overwhelming already here, right? As I'm scrolling through this, there's lots and lots and lots of information to sort of parse through and understand. One thing you may notice as you are looking at this is there are quite a few rows in this data that appear actually quite similar. And there may not actually be an important distinction between them in all cases. But at the same time, there can be individual rows out of these thousands that may be the critical row for this experiment. So this is really the challenge that is motivating our work here, right? Can we somehow aggregate these similar rows together while still preserving these small but interesting details? And to start, I'll talk sort of at a high level for how we have approached this with our visualization. And I'm going to break this down into 4 steps. First, we sort our data. This is a very abstract representation of data because you can have different structures for data and it should still apply as long as you're able to define a similarity between your rows. So if you define somehow the similarity of your rows of data, you can then sort your data so similar rows are next to each other. And in the same vein, you can also group rows together that are similar, right? And in practice, the way that we do this is we use hierarchical agglomerative clustering with this similarity metric. This gives you this dendrogram or tree. Once you have that tree, you can use it for your sorting. You can use it for your clustering. So once you have your groups, you can summarize them together to get sort of a summary or representative row for that group. And this is really however you choose to define this, right? For numbers, that could be the total or average or some other fancy function. For collections of categories, you can return the most frequent or the frequencies of all of the categories in that group. And then the final step here, once you have your summary representation in data, you need to visualize that in some way. So depending on your use case, you can encode this as color, size, and so on. So that's sort of the high-level approach that we take. For the rest of the talk, I'm going to go through a real use case example with this aggregate heat map that we've developed. And for this, we're going to be looking at real data from this paper. And in this paper, the authors collected samples from healthy tissue as well as tumorous tissue. And what they found were 10 phenotypes where there was a big difference between these two groups. So when you load this data into the OZ system, you get roughly 500 phenotypes here. And I've highlighted 3 rows here that are these flagged interesting rows. You maybe can't see, but they have this tiny green mark next to them. And for these 3, the phenotypes are overexpressed in the tumorous tissues. So one thing you may be wondering throughout this is, why don't we just show all of the data? And you can, right? This has every single row of data in our data set just squished together. But even up here, right next to the projector, you really cannot distinguish those individual rows that are green and marked as interesting. But with our technique, we can, based on how we define our metrics, we can group all of those uninteresting rows together, create that summary representation, and that leaves a lot more space to see these few rows that are flagged as interesting. And the advantage of this is that you get a little bit more context here, right? Here we see our interesting rows with this uninteresting row. It was not flagged, but it actually appears very, very visually similar. Yeah, and then for the rest of these slides, really the point is that this system is very flexible, and you can take this aggregation to the extremes, right? Here I've aggregated all the way down to these 2 rows. Here amongst these interesting phenotypes, there's really 2 groups, those that are overexpressed and those that are underexpressed in the tumor. And if we bring this back to where we started, this really illustrates what I like about our approach. It's really quite flexible. You can apply it to different data. And you have full flexibility from showing every single row here all the way down to this extremely condensed version. So with that, I'll open it to questions. Thank you very much.
Hi, thank you. So for your methods, how do we define these interesting rows that you're talking about on a stereotypical RNA sequencing experiment, single cell? Like, are these the genes that are significantly associated with our disease or condition? How would you go into a more, let's call it like stereotypical single cell experiment?
Yeah, so the question I heard was, how do you define what is an interesting row? So to give the concrete answer for this experiment, what I did was looked at those phenotypes that the authors defined. So this is kind of a proof of concept in this case. But you could do something like you're describing for these 2 conditions, look at among these 500s, which are the most statistically significantly different, and come up with some test that flags that. Of course, then you need to consider how, what are your 2 groups that you're defining, and not necessarily a trivial thing, but you could come up with some type of system like this. Yeah.
Got it. Thank you.
Next.
Yeah, thank you very much for the interesting talk. I'm curious, you said you sort them by some sort of similarity. Can you maybe say which similarity measure you use in the examples you showed? And then also which rows you're choosing is kind of like the representative row after you boiled it down to this simple overview.
Yeah, okay, so there's 2 questions. I'll start with the similarity one. Let's see if I have a better screenshot. Yeah, maybe here is a good one. And our system already has a few options built into it, but at a high level, there's sort of 2 important pieces per rows, right? There's the markers, and there are the cell counts, we'll call them. And among 2 rows of cell counts, think you could do something like I think we have like a Euclidean distance or like a cosine distance, this type of thing. Although if you have distance, you have a similarity sort of trivially. And then the second piece are these markers. Again, you can use some, like, I think we use a Hamming distance, basically how many proteins are different, right? And then you can combine those two distances, assign weights to each of them, and then you have a very flexible sorting, right? I want to sort entirely by the protein markers, or I want to sort entirely by the cell counts, or like some nice combination. And then your second question, remind me your second question.
Yeah, you couldn't collapse them together, right? And I was wondering, what are you showing then in the end? Like, are you combining the rows, or are you choosing a representative row for the group?
Yeah, again, here you have some different options. I think for us, right, like in the rows, you have these cell counts. I think we add them together so you get the sum, and then you can show the cell counts. I'm skipping some details. Actually, these are not mapped to cell counts. It's like statistically transformed, and then you have the z-score. But conceptually, that's what we are doing. Yeah.
Thank you.
I have a simple question. How did you come up with the color palette?
Great question. I think I inherited that when I started the project, but we have this, I think this is commonly used. So I think this is familiar to people where at least our users, where the red is overexpressed and blue is sort of underexpressed. And the white is like middle of the road.
Yeah, thank you. Let's thank our speaker once again.